Hey everybody, sorry I'm not here today. I am going to be out today and tomorrow, so we're going to talk about cell division. We will probably talk about cell division today and tomorrow, but I just wanted us to get started. So if you labeled your cell on Friday, be sure that you keep that because you will need to study that for your test. And don't think I did that animation that's on there because I'm not that smart. Someone else did that. Okay, so we're going to talk about cell division or mitosis. Reasons for mitosis, there are three main reasons: growth, repair, healing, and asexual reproduction. Our cells have to reproduce so that we can live. Cell division by mitosis, it takes some cells divide constantly. Cells in the embryo, cells in the skin, cells in your gastrointestinal lining. They are always replenishing. They are always your blood cells. They are always reproducing so that we can continue to grow. Epithelial cells, if you'll remember, those are your skin cells. Interstitial cells, those would be the cells in your GI tract. And then that was an embryonic cell. That's an embryo, a 7-week-old embryo at the top. Cell division, other cells divide rarely or never. Things that don't divide, that just divide rarely once it's complete, are your cardiac cells. They rarely repair. Your spinal cord and nerve cells, once you have all of those solidified and your brain becomes non-elastic after about the age of 11, your brain is what your brain is. You can improve synapses, but you can't grow new nerve cells in the brain or in your spinal cord. So remember that our skin, our blood, our GI tract, it's always, we are having some continual reproduction there, but our nerve cells, our heart cells, some of our major organ cells, they don't replenish the way our skin cells do. There are two kinds of cell division: mitosis, which is a somatic cell, and then meiosis, which is the creation of a new sex cell. So new sperm or egg cells, which we don't really make a lot of new egg cells because most women are born with all of the egg cells that they're going to ever have. But we do have our mitosis is going along all the time. Somatic cells has a cell body. And it will be whose genes will not be passed on to future generations. So the DNA that's in that our cells replicate, but they are an adult cell basically, somatic cells. Germ cells, those are cells that are destined to become a gamete, a sperm. They're going to become sperm. They're going to become egg. Those are genetic cells. They have your chromosomes in them. And they are going to be passed on to your future generations. So you will pass those down to your children if you have them. Cell division, things to know, the good vocabulary to remember for cell division is diploid. That is a cell with 2 chromosome sets in each of its cells. And all of them are body cells. Sorry guys, I didn't mean for my phone to go off. And then a haploid is has a single set of chromosomes. So that's like your sperm and your egg. So your adult cells, your normal body cells are diploid cells, and then your sperm and egg, those are haploid cells, or N1 and N2N. Characteristics of mitosis. A diploid cell will give rise to another. So one diploid cell is going to make another diploid, however you guys want to pronounce it, cell. The chromosomes numbers remain the same. DNA remains identically the same in every cell. And one cell gives to 2 cells. So once again, and those are going to be that same cell is going to regenerate itself. So the cells on my skin are going to make the same cells that were already there to replace them. Same thing with the inside of my mouth, the mucosal lining on the inside of my mouth. Those cells are going to regenerate over and over as the same cell. it has all of the DNA. It has all of the DNA that my body has. So it has 23 sets of chromosomes, or 46 chromosomes in each one of my cells. The cell cycle, a typical cell goes through a process of growth and development and reproduction called the cell cycle. Most of the cell cycle is called interphase. Now, I know this is a review for a lot of you guys, but cell division is important in anatomy specifically because we are studying cell regeneration. We are studying the body consistently, and our body is pretty much either regenerating or repairing at all times. The longest phase of the cell cycle is interphase, and the three stages of interphase are G1, S, and G2, and we'll talk a little more about those. So the S stage is where DNA is synthesized, but we'll talk some more about that. So this is just a bigger picture so you can see that interphase is the S phase, that is the growth and DNA replication. I'm sorry, I should have started with G1. G1 is the actual growth stage. The S stage of interphase is growth in DNA replication, and G2 is the final preparation for the division. So it's as you know that when we talked about before how those chromatids, they separate and they go to the end of the cell, and then DNA replicates and it makes two new nucleus and they start to separate apart. So cell division, all living cells come from other living cells, so our genetic makeup comes from each. It separates out and makes two new cells. During mitosis, the nucleus of each cell divides, forming two nuclei that are identical genetic information. Now, what happens when our DNA doesn't replicate the way it's supposed to? We have what's called a mutated cell. We can think about different kinds of cells being mutated over the course of our life. Some of that might be that we know some of that is cancerous cells, or we know them as non-malignant tumors. Sometimes there's something called a fibroid tumor. It's an excessive growth of cells that aren't really complete. Another way we think about a type of cancer this is really specific to would definitely be like a leukemia, where the cell doesn't divide appropriately, it doesn't mature appropriately. So those are some definite issues where cell division is not an exact replica of the previous cell. Mitosis produces two genetically identical cells. I know this is really kind of repetitive, and it's kind of rudimentary for you guys, but I don't know what you guys learned in biology, and it's been a while for you guys. So we have the parent cell that goes through prophase, and then metaphase, anaphase, and telophase. So you guys will need to know all of those and remember all of those. So in prophase, the cell begins the process of division. The chromosomes condense and they get shorter to make those chromatids. Then DNA wraps around the chromatin, and it folds and makes the chromosome. So it makes the duplicated chromosomes there. So it starts out as DNA over here on the left. You have your GC and your AT, and then it starts to condense and get smaller so that it can get into the form of those chromatids. The nuclear envelope disappears during the prophase, so the nuclear membrane disappears during prophase. So you just have the nucleus itself there. The centrioles migrate, so they separate, and they go to each end of the cell. The centrioles, we talked about that when we were talking about the cell the other day. To each end of the cell, and then those spindle fibers start to form. So that's kind of a drawing of prophase. I want to give you guys 3 minutes because it's important to know what it looks like. So I want y'all to draw prophase. So I'll go back here and let you look at this image. So the nuclear membrane has disappeared. And that's what prophase looks like with all of the pieces in it. So I want you to look at this picture, and I'm going to give you about 2 minutes, and I want you to actually draw prophase. Just because I'm not there doesn't mean you don't need to be drawing it, guys. I'm going to look at it when I come back. Good to have these images in your notes for when you study, and hope you guys are taking Cornell notes, because I'm going to look when I come back about with them to see if you guys are actually doing this. Got about 30 more seconds. Okay. So that's a kind of a more clear picture of prophase, where you can see the spindle fibers and the centrioles and the aster there, and the centrosome there from the original cell. Because remember, we said the centrioles are housed in the centrosome, so they are. So that's another good image of prophase. Metaphase, the chromosomes line up at the equator, or the middle of the cell, with the centrioles at the opposite ends, and the spindle fibers attached to the centromere. So you can see there are your chromosomes there in the middle of the cell, and your centriole down here at the bottom, and your centriole there at the top. The spindle fibers that are separating out from each end. And these are just some good microscopic images of what's going on. There you can see there's the chromosomes there in the center of the cell. Alright, I'm going to give you guys 2 minutes to draw metaphase. And I'll actually set the timer this time. Set a timer for 2 minutes, please. Set a timer for 2 minutes, please.
Okay, 2 minutes of counting.
I know that cracks you guys up. I want you to have good images in your notes for you to study. We got about not quite a minute left, about 50 seconds left. So we're going to talk about anaphase now. In anaphase, the centromeres divide, and they start to move towards the centromeres that were here in the center. They start to divide, and they go towards each end to their sister chromatid. They separate and start moving towards each end to start that complete cell division. The spindle fibers contract, and the chromosomes are pulled to the opposite end. So those chromosomes that were together now are separated, and they're moving to the ends of the cell. All right, I want you to draw anaphase. What's going on in anaphase? That they are those chromospheres are moving away, moving towards those spindle fibers and the chromatids, towards their sister cell, their sister chromospheres. Okay. So telophase is actually when the nucleus, in telophase, the nucleus actually divides. The chromosomes are at the opposite ends of the pole, and the nuclear envelope or the nuclear membrane starts to reform around those chromosomes, around that nucleus again. So now you have these two nucleus, one at each end of the cell, and that the nuclear membrane has started to develop around it. Alright, now I want you guys to draw telophase. And I'm being really specific about this because one of the things you guys will have an opportunity to do on your test, you will either be able to draw or you can list and describe what happens in cellular division. That's going to be a bonus question for you guys. And if you can draw it and label it, that's the same thing for me as describing it. But you got to label it also. Okay, give you another minute to draw it. Okay, so the last thing that happens is cytokinesis. what you're looking at there is a frog sale. Cytokinesis is actually the cell completely dividing into two separate cells. So that's what I want you to remember, that cytokinesis is the development or the actual separation into two daughter cells. And like I said, you can see that crease there where it's actually starting to split apart. So you have interphase, which we didn't talk about, but interphase is just your normal cell. Interphase is just prior to anything happening to that cell. It's just your normal skin cell that's lived about its 26 or 7 days. Then the cell oh, sorry guys. Oh, I meant to go backwards. Then it goes from interphase. What is the next thing to happen? You have interphase. You have prophase, right? Next, after interphase. Then you have metaphase. Then you have anaphase, and then you have telophase, and then you have cytokinesis, where you have two separate cells. Like I said, I know that was really elementary for you guys, and it was very specific, but I want you guys to have a good understanding of how the cell actually divides, because you're going to be looking at multiple different kinds of cells over the over the year as we go through different systems and all that. So you're going to actually see different types of cells. So I want you to have a really good understanding and a really good review of what the different parts of mitosis. All right, guys. I will be back on Tuesday. You guys will have, we're going to talk some more about cellular reproduction tomorrow. If you have questions or you need anything, just leave me a message on Classroom. So if you'll look in your classroom and you have questions, you can comment or question. Just remember that if you leave comments or questions that are inappropriate, that is available to administration as well. But if you're having trouble with something, you have questions, you can message me privately, or you can post public messages on Classroom. You do have that opportunity. The other thing I want to remind you guys is be sure you're checking your email. Every day when you come into class and you get a Chromebook in my room, you need to check your email. It's at the bottom. It's a little red and white envelope. Just click on it and check your email to see what email you're getting from me. That's how I'll return your grades to you on quizzes or anything like that. And I ask you if you want a printed out copy of any of the correct answers of your quiz with the correct answers. But unless you respond to me, I don't give that to you. So be sure, and I'm pretty sure you're going to see something about this tomorrow, maybe in the quiz form itself. So you guys have a good day. Please take care of your sub and take care of each other, and I'll see you on Wednesday.